Video is to help you learn solutions to bacterial ruler problems. Over time, your goals are automaticity, in other words, automatically in a biological image, seeing what's what, knowing what you're looking at without having to think too much about it. That's related to the process of developing an understanding of image analysis for any kind of biological imaging, including the kinds of imaging that we do in medicine. Second goal is getting a quick automatic understanding of how big, how small, and how far things are in these images, and then also how much, how many, and how fast. This first image shows macrophages. This is part of a macrophage cell. These are immune system cells. They're amoeboid. They move around through the body, but you don't need to know that to analyze this image. And in this particular case, I'll tell you now that they have phagocytosed, ingested a bacteria from the stomach, Helicobacter pylori. That's these black things. This one, C, is a close-up showing 4 bacteria, these ovals that have been phagocytosed. And this one is a portion of the whole cell showing one big mass of bacteria that have been phagocytosed. So let me walk you through how I would analyze this. First, in these images, what structures and organelles can you identify? Well, I've told you there are bacteria that have been phagocytosed. The first thing I would look at beyond that is there anything I would consider to be a membrane? Membranes are continuous dark lines. So anytime you see a continuous dark line surrounding something or along the edge of something, that is most likely to be a membrane. So we've got some candidates here. We've got black lines here. They sort of fade in and out of grays and blacks, but you can see a continuous black line running along here. There's also a clearer black line here surrounding these bacteria. Again, it kind of fades out in a spot, but looks continuous all the way along here. There's some others you can see. Here's another one here. Here's a couple here. So those continuous lines are membranes. So that already tells us we've got something from the bacterial ruler. We've got membranes, and that means something about that thick, about the same thickness of that line, is going to be approximately 7 to 8 nanometers, or if you want to round up 10 nanometers. So that's the thickness of that line, the thickness of these membranes. What else can we identify? There are these membrane-bound structures that I hope you can see a hint of some parallel lines. I'll sort of indicate them. There's one here, a dark line, another one here, and maybe another one there. Same here, this one. There's a membrane around it, but there's also some dark lines here and here. Anytime you see something that has a series of approximately parallel wavy lines, that is almost certainly a mitochondrion, because the structure of a mitochondrion is to have an outer membrane and then an inner membrane that does this. And so when you look at them, you see these stripes from these membranes going across. So that's probably a mitochondrion. This is probably another one. Here is probably another one. You can faintly see some lines. Now these take practice to identify quickly, but over time you should get better at doing it. In this image, about the only things I can tell are the mitochondria. This is a vesicle. This is the cell membrane, and this is the this is the membrane surrounding the vesicle that has the bacteria in it. These might be endoplasmic sizing. I'm guessing that they probably are. I can't see anything that I clearly see as ribosomes, partly because at this scale, the image is a little bit fuzzy. Let's look at the other image. Now here, this is an entire bacteria. Here the bacteria are like this. By the way, you're seeing some of them as circles and some of them like this. Helicobacter pylori are actually spirochetes. They're spiral shaped. If we looked at one sideways, it would look more or less like this. And so when we cut them, we see some as partial curves. These ones we've cut, they're going in and out of the image. And so you can't see the part behind. This is just a thin slice. Can we see anything in here, anything else in here that we identify? Well, these bacteria are all in here. These may also be bacteria. They're pretty dark. And so that would be my guess. They're also bacteria. Up at the top here is a boundary. I'm seeing, I hope you can see this dark line along here. And that is a fairly thick line. It's probably the double membrane around a nucleus, but at this magnification, you can't see the space between, and so it just looks like a single solid line. The other thing that makes me think this is a nucleus is that there's a part of it here that is darker and part of it that is not, and that would be the parts of the nucleus that are darker and lighter. Again, I can see something that looks like it's around these cells. That could be a membrane. It's a little fuzzy, but it does surround things. So that would be my guess also, is that that's a membrane. I've told you that these are from the same cell, so that this is a close-up. So what structures can we identify? We've got mitochondria here. We've got the cell membrane, the plasma membrane around the cell, and the vesicular membrane. We sort of hazily see the inner and outer membrane of these mitochondria, maybe some ER, endoplasmic reticulum membrane there. And then, of course, the bacteria. On this one, my guess is a nucleus. It's an informed guess. I'm not absolutely certain. I'm not sure that other than this vesicle, I can distinguish much else in this image. There is an outer membrane here, but at this magnification, I can't even see it. This just looks like a uniform dark gray until it gets to the white gray outside the cell. I've walked you through some of the evidence that supports the identifications, and you should think through the same things for yourself. Question here, and see how wide is the vesicle holding the 4 bacteria. It's the distance between the white bars. So here's one white bar, here's the other. So what's that distance? Well, in any bacterial ruler problem, if you can find something you're pretty certain is a membrane, that is going to be one of your best measures. Of course, for here, that's pretty small. But that's a membrane. So let me put some red bars here to represent about the thickness of the membrane. I'll offset them slightly so that we can count them. And what I've got here is about 10. So this whole thing, oops, let me try a different color. So this length might be about 10. I think I've actually got my lines here a little thicker than the membrane actually is, but it's going to be close enough. So 10 times 10 nanometers is going to give us about 100 nanometers. So that distance is 100. And once we have that, we can just put marks across it roughly that same distance apart. And we have 1, 2, 3, 4, 5, 6, 7 of those. So that makes our answer about 700 nanometers, which would be equal to 0.700 microns or micrometers. When you're doing bacterial ruler problems, if you have something that's really small, really thin, and you're trying to use it to measure a large distance, the best way to do it is to get an intermediate measure, in this case about 10 of these, about 10 membranes, and then go from there with a series of markups. How confident am I in this number? Well, I'm pretty sure that this is at least 500 nanometers, and I'm pretty sure it's not over 1,000 nanometers. That's within a factor of 2. That's usually something we're pretty happy with. So I'll go with my 700 nanometers or 0.7 microns. How can we check? It's handy to have something to check if you can. Well, we've got bacteria in here. So we've got a width of bacteria and then, of course, length. Well, width of bacteria is, at least of an E. coli, is about 1 micron. So if that's 1 micron, then this whole thing would be about 1, 2, 3 microns. So using the bacterium, although it's not an E. coli, that would give us an alternative of about 3 microns. And that logic would the logic of either one of these would probably be acceptable, but you would have to show your work. Which one is more reliable? Probably this one, using the thickness of the membrane. But the image is a bit fuzzy. The membrane may be showing, because of the fuzziness, it may be appearing to be wider than it really is, in which case maybe the right answer is approximately 1.5 or 2 microns. Again, show your work, and all of these would probably be in a reasonable range for an estimate of size here. In D, what is the width of the entire bacterium? Well, here the best measure we have, if we don't use anything from this, is going to be these bacteria. And your first guess for a bacterium is going to be a width of about 1 micron, equals 1,000 nanometers. And length, depending on the bacterium, can be quite different. Now, bacteria vary in size. Some of them are huge, but most of them would be in this size range, and it's a reasonable first guess. So that gives us this distance is about 1 micron, or here, 1 micron. So if we start putting marks across, notice I'm not trying to be perfectly accurate, but I'm trying to get them to be about the width. And now count. What do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's about 10. And that's about another 10, and that's about another 5. So I'm going to go with 10 plus 10 plus 5 is 25 microns. Again, if I show my work here and I point out that what I've done is estimate size based on the width of this bacterium, that fits. We have one other thing in here that could help, which is this nucleus. Now, we're only seeing a part of it, but if this is the nucleus, it's pretty long. This nucleus would be 15 plus a little bit perhaps microns based on our estimate here. And that would be pretty large for a nucleus, but two things: nuclei vary in size, and plus they're not always entirely spherical. So if this nucleus is flattened, it wouldn't be surprising if it was that size. But one of the things it suggests to us is that maybe this is somewhere between 5 and 10 microns. Maybe the real size is about 15 microns, and these bacteria are a little less than 1 micron. It doesn't matter. That would also be within our factor of 2 range. Another question. These images are digestive enzyme-secreting cells of a simple tablet animal. It's in the phylum Placozoa. They are very, very simple animals, among the simplest, apart from sponges. And but nonetheless, they have cells that are a lot like other cells that of animals. So in image 1, this one, what structures and organelles can you identify? So we have to zoom in a bit. And here I'll let you stop the video if you want to see what you can see before I comment on them. Your best prediction for these, your prediction for this, your prediction for these long lines, your prediction for these. By the way, I am not sure what they are. Probably secretory vesicles. And some of the finer scale things are hard to see. There looks like there are some lines in here that might be some other structures, but I can't make them out well enough. Okay, so what do we have? First, these look like mitochondria. Notice the lines here. They're usually squiggly, but some of them are running parallel to each other. So that's the outer membrane of the mitochondrion, and then we have the inner membrane going like so. So those are mitochondria. This is the cell membrane, the plasma membrane between cells. We're looking at here the boundary between two cells. You can't be certain of this here, but this white space between cells often is just the extracellular fluid between cells, and you can see a thin black line that is the cell membrane. And so where you see two of them with varying sizes, that's often the boundary between two cells with this light space between them, but not always, because here is a vesicle inside, and then here's the membrane of the cell. So this is the membrane of the vesicle. This is a nucleus. You can, in this case, see the double membrane. Here's the inner nuclear envelope, nuclear membrane, the outer together making up the nuclear envelope. And a clue that it's the membrane is that some parts are darker and some parts are lighter, and that corresponds to how tightly the DNA is packed. Heterochromatin and euchromatin, showing this modeled pattern of darker and lighter parts there. There are some other vesicles that are a little bit faint, and I'm not sure what all of those would be. I'm not sure what vesicle this is and what exactly is inside of it. I can't tell from the image. It could be enzymes that are being secreted, have been packaged in there, are going to be secreted out the bottom of the cell. Back to a ruler problem question. What's the length of the cell? So going from here, this looks like the top of the cell. Here's the next cell up above it. So from there, all the way down to here, our cell looks like it ends down here, and then this is part of another cell down below. So what clues do we have? Well, we have the membrane, and we could use that. We also have the nucleus, but a problem with the nucleus is if you have a spherical nucleus and you cut through it in the middle, you'll see the whole nucleus. But if you cut through it at the edge here, the nucleus will appear much, much smaller, only this large. So nucleus, the upper bound is probably about 5 to 10 microns, but the lower bound is hard to tell. Mitochondria are helpful, although they vary in size in different species and even in different cell types. But mitochondria are often about 0.5 to 1 micron, though they can be smaller or larger in diameter. So what do we have so far? Well, we have this might be 5 microns if it's through the center. These might be 0.5 to 1 microns. The membranes are still our most reliable guide, although in this image they're small enough and fuzzy enough they might be tough, but I'd still make an attempt with those. So at this magnification, here is roughly the thickness of one membrane. My pen is probably a bit thick, but not too bad. So let's take something else, part of this mitochondria in here, and put some lines in that I can count. Stack them up here, and that looks like about right. So what do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 lines. So 10 times 10 nanometers equals 100. If you want to be a little more exact, you could do 10 times about 7 or 8 nanometers equals 70. But now we have something bigger we can use as a unit. So let's take that and get roughly the size of this nucleus. So there's 1. That distance looks about the same as this distance. Going to be close enough. Here's 2, 3, 4, 5. So add them up. 1, 2, 3, 4, 5. I get this distance is somewhere around 5 microns. Whoops. Let me back up here. These were only 70 or 100, so checking my work, I get this distance is about 5 times 100 would be 500 nanometers. Or if I'm doing this one, it would be about 350 nanometers, which suggests if this is the nucleus that this might be not through the center of the nucleus, and that these are relatively small mitochondria. Let's see what measure we get using this. And now let's go from the top, and we're going to use this measure. I'm going to switch to black. Here's 1. Here's 2 of whatever those are, nuclei, 3, 4, 5, 6, 7, 8, and a bit. So 8 times 500 equals 4,000 equals 4.0 microns. That suggests the whole length is about 4 microns. That is possible for this cell. It seems a bit small to me. Eukaryotic cells are more often in the range of 10 to 20 microns, and 20 microns being a more common average. And the fact that this looks like a nucleus, suggests that my estimate here may have been a bit small. So I might say, okay, based on membranes, I'm going to go with 4.0 microns. Based on the nucleus, if this nucleus is 5 nanometers, then that would suggest the whole thing is 8 times that, then 40 nanometers. That seems pretty big for this cell, and that would suggest these mitochondria are pretty large, but it's possible. So those would be the range of my estimates. The smallest would be 4 microns. The largest would be up around 40 microns. And I might pick something in between. When I have 2, I might say, okay, I've got 2 estimates. I'm going to go with 20 nanometers, 20 microns, the amount in between. I'll make a comment here. If this seems a little fuzzy to you, that is part of what this whole exercise is about. You're making estimates. You're not trying to have it be exact. And you want estimates you can defend. And so showing your work means you can defend the answers that you gave, and then you can also check them to see if they're logical. You want to be able to do this quickly, and so practicing bacterial ruler problems will help this become more automatic. In image 2, what structures and organelles can you identify? Well, membrane, 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 more membrane over here. Then we've got some curved consecutive membranes, and that to me immediately suggests Golgi apparatus. And remember, the Golgi apparatus secretes materials, puts materials into vesicles. That might be something that's secreted outside the cell. It might be new membrane and membrane proteins that will just fuse with the membrane of the surface of the cell. Can be materials that are going to be sent to fuse with a lysosome or sent somewhere else. So this looks like a secretory vessel of Golgi. So Golgi here, secretory vessel of Golgi. And then this might be a larger secretory vessel. So this is one that's still being filled and forming. This one might be a full one, and it's near the membrane. Maybe it's about to be secreted, released, or maybe it's waiting for some chemical signal to be released. This darker stuff is cytoplasm. You can see some smaller membrane materials that might be just cut through parts of the Golgi. So if you did a cut through this part, you would get a circle. There's not much else I can identify. These have the sort of gray look of mitochondria, but they're way too small if this is a membrane. So what's the size of this? Again, I'm going to mark it up. Here is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 20, 21, 22, 23, 24. So about, call it 20 or a bit more times the width of a membrane. 20 times 10 would be 200. 20 times 7 would be 140. So I'm going to go with either one of those and call it about 140 to 200 nanometers. If you want to count this as 25, maybe instead it would be 140 to 250 nanometers. What material does this contain? Well, it's from digestive enzyme secreting cells of these tablet animals, placozoans, and that suggests that these might be digestive enzymes that are being packaged for secretion. Last one. This image is from the excretory organ of an annelid worm. It is analogous to mammalian kidneys. They collect material that needs to be excreted, waste material, and send it out. The way it works is this is extracellular fluid of the annelid, and it contains valuable molecules like glucose, amino acids, sugars, and so on, and proteins, as well as waste molecules, such as urea or uric acid or others. And it is going to go into the tubule. This is the lumen, or the space of the tubule. And so the waste molecules are entering the tubule through these little gaps. All these arrows are showing gaps, tiny pores that material can enter. The valuable molecules can enter along with the wastes, and so as this material moves down the tubule, there are cells aligning the tubule that reabsorb, take out the glucose, amino acids, and other valuable molecules. So the waste and glucose get in, but as you get down, only the wastes are left. The glucose and amino acids get taken back out. Okay, what can you identify in this image? And here I'm getting you to think about with confidence and with an informed guess. You don't know the scale yet, and so all you have are these continuous lines. Continuous lines are probably membranes. So these seem to be enclosing here, which makes sense because there are little pores here. And so this would be like a sieve with little holes. It's a thin section, but if you looked at this, turned it on its side, it would have lots of little holes, which are all these gaps. Okay, so what can you identify in this image? You can identify the membranes. There may be or may not be a hint of another membrane here. It's hard to tell. These are membranes, and so these are presumably, this is extracellular fluid, this is fluid with wastes and that is entered, and so all you really have is these membranes. This looks vaguely like it could be a mitochondrion, but it doesn't really have the parallel lines we might expect. It's probably some structure that's just part of this cell. So with confidence, the only thing I can identify is these membranes. This might be a vesicle, but it could be. It's pretty small, but there are some vesicles that size. There are some dark dots here, which in other images I might think were ribosomes. And it's possible, I'm guessing here, it's more likely something else, but they could be. But let's think about it. If this is a membrane, then that is about 7 to 8 nanometers, or we'll round to 10 nanometers. If that's 10 nanometers, that line, then these cannot be ribosomes, because ribosomes are bigger. When you see them along with membranes in the same image, what you'll see is the ribosomes are about 2 to 3 times as thick or wide as membranes. They're about 20 or 30 nanometers. So with confidence, pretty much just the membranes. Approximately how wide is the opening at A, that opening there? From the looks of it, I would say it's about 1 to 2 times the width of the membrane. And so that would make it somewhere between 10 to 20 nanometers. So approximately how wide is it? Thinking about it here, I'm actually looking at, it does not look like it's 2. I'm going to go with 10 nanometers. So 10 nanometers, about width of membrane. So what could or could not pass through opening A? Waste molecules such as proteins? Maybe. Small proteins, potentially. Big proteins, no. If we've estimated it correctly, even medium-sized proteins at about 10 nanometers, medium to largish, would be able to go through. So we can tentatively put it down as a question mark. Waste molecules the size of monosaccharides? Yes, for sure. Waste molecules the size of disaccharides? Yes. Those are down on the order of 1 nanometer or a little bit less. Water, of course, so yes. Ions, yes. So it's going to freely allow these waste molecules through, along with sugar, amino acids, and ions. Large proteins won't get through. Smaller proteins, maybe. In fact, these are actually small enough in reality, we can't tell it from this image, but they're small enough in reality that only very small proteins will fit through. All right, I hope this helps with doing bacterial ruler problems, and if I'm asked, I will post more.